Okay, are there any questions? Everybody's good? All right, let me share the screen. So today we're covering Host Defenses 1. Note that there will be a quiz this Friday on Chapter 7 on the lesson on antibiotics and on Lab 7. Any questions about that? So Lab 9 and Host Defenses 1 will not be on the quiz this Friday. Any questions about Lab 9? We'll meet for the lab later today, but actually it's only time for you guys to do work. There's nothing we'll do today in the lab. All right. Can you see this?
Yes.
Thank you. So I believe we stopped at the second line of defense. Let me just do a little bit of review and say that we're going to talk about the second line of defense today. It's a non-specific defense. Yesterday we talked Yesterday we talked about the first line of defense. It also was a non-specific defense, and the first line of defense was mainly barriers to infection, such as your intact skin and the intact mucous membranes and the secretions of your mucous membranes. And we're not really going to talk about the third line of defense today, although there is one aspect of it we'll talk about. It works with the second line of defense. But we'll talk about this in our next lesson, the third line of defense. That's a specific defense involving specialized lymphocytes, mainly the B and the T lymphocytes, and then the antibodies produced by the B cells, the B lymphocytes. So we talked about the skin last time and how it's an inhospitable environment, salty, dry, slightly acidic, and very things can get through the intact skin. Most things that get through the skin get through when the skin is damaged. We also talked about the mucous membranes and found that they were not as good a barrier as the intact skin. There are a few things that can get through the mucous membranes, and as you know, colds and flus and a number of the other respiratory ailments, and then the sexually transmitted diseases get through the mucous membranes. All right, so let's talk about the second line of defense. Sorry, I've got a frog in my throat for some reason. The second line of defense is still non-specific, and it involves the phagocytic white blood cells. And when we're talking about white blood cells, I need to switch my glasses. The white blood cells are often abbreviated WBC. So if you ever see that, that means white blood cells. The second line of defense also involves inflammation in the antimicrobial substances like interferon and the complement proteins. And we'll talk about all of these today. Let's first talk about the phagocytic white blood cells. The white blood cells and red blood cells originate from the same stem cell, and that's a stem cell in the adult bone marrow. In very young children and in fetuses, that can happen in a liver stem cell, but in adults it happens only in the stem cell in the bone marrow. So let's watch a little
Blood
video.
cells originate from stem cells.
Can you guys hear that?
Yes.
Blood cells originate from stem cells in bone marrow. Red cells are produced by a series of changes in stem cells, in which the cell nucleus is lost and hemoglobin accumulates. The red cells then enter the circulation. The blood-clotting platelets, or thrombocytes, are fragments of cytoplasm from the stem cells. White blood cells, basophils, neutrophils, and eosinophils all have segmented or lobed nuclei, while monocytes and lymphocytes have single, more spherical nuclei.
Okay, any questions about that video? So if you've ever looked at a blood smear or looked at human blood, or actually most blood of mammals, you note that most of the blood cells are red blood cells and they lack a nuclei. And we're not going to talk about those because they don't defend the body. They supply oxygen and nutrients, but they derive from the same stem cell in the bone marrow. And that's the erythroid line of the blood cells. In the myeloid line of the blood cells, we get the phagocytic white blood cells. You also get from the myeloid line granulocytes, which create the platelets, and we're not going to talk about those today either. Now, there is one other white blood cell we need to talk about it, and it comes from the lymphoid line of the blood cells, and it creates the lymphocytes. And when we're talking about the white blood cell, shown here, it's these cells, the basophils, the neutrophils, the eosinophils, the monocytes, and the lymphocytes. And these are the cells you see in the blood that are not red blood cells. Any question about that? All right. And sometimes they have this picture a little different that they'll have. I think it's these two are on the same line for a while, and then they split into the myeloid line and the erythroid line. And how the graph is, it doesn't really matter, because we're only going to look at the cells here. The lymphocyte we'll talk about because it is a white blood cell, but realize we're really going to talk about this in our next lesson because it's related to adaptive immunity, and it is, oh, there we go, the leukocytes. Those are the white blood cells. We'll talk about that in our next lesson. We'll just briefly talk about the cells today, but we'll spend the whole lesson talking about the lymphocytes tomorrow, I guess. And we'll talk specifically about basophils, neutrophils, eosinophils, and monocytes right now. And we will talk a little bit about lymphocytes. We can break the white blood cells into two groups, the granulocytes and the agranulocytes. And that's not shown here. Let me see, there's a picture that shows it. Oops, well, that's not good. Let me see if I can find it. I thought I tested this and got all the pictures to work. Yeah, that should work. Why isn't that working? Oh well. Let me think and see if I've got another picture. Let me open this up. Just see if there's a picture here that works. Yeah, it's there. I'm not sure why that picture isn't coming up I'll look at that link and try and show that. Figure 15.5. And these figures go to another textbook than the textbook you have. Let's blow that up a little bit. So the granulocytes are the three on top, and they have granules in the cytoplasm as well as in the nucleoplasm. Let me think about that. The agranulocytes don't have granules in the cytoplasm, and that's just dividing up the white blood cells by the granules. Okay? Any question about that? So the granulocytes include the basophils, the eosinophils, and the neutrophils. Let's talk a little bit about the basophils. They stain purple when you stain them, and the basophils are a very rare cell in the blood. 0.5% to 1% of all the white blood cells in the blood, so they're very rare. And they have a, what do you call that, binuclear nucleus, meaning it's split into two parts, and it is connected together, although there's not much of a connection. Anyways, that's the basophils. And they release histamine and other substances important in inflammation. And they're important for allergic response and then starting inflammation. Any question about the basophils? And you can pronounce that basophils also. Okay? The next white blood cell, which is also granulocyte, is the eosinophils. And they're a multiphagocytic cell, and they stain reddish when you stain the white blood cells. Their numbers rise during allergic reactions, so they're involved in the immune response of an allergy. And they also rise, meaning they're more commonly found during a helminth infection. So when you have a worm infection, you'll see much more of the eosinophils. And even though they are multiphagocytic cells, that's not how they attack a worm, because the worm would be too big for an eosinophil to phagocytize. When they're attacking the worm, they come up and bind to the parasitic worm, and when they attach to the surface of the worm, they secrete peroxide ions externally to the eosinophil, and the peroxide ions come up against the cells of the worm, and then it kills the cells of the worm. And if you have enough eosinophils coating a parasitic worm, the worm can die because so many of its cells are killed by the eosinophils. Any question about that? And they're also a rare cell in the blood. Normally, they're only 2 to 4% of the white blood cells in the blood. But during an allergic reaction or during a helminth infection, you'll see that number elevated. Any questions about any of that? And so clinicians, when they suspect a patient has a worm infection, something they can do is just take a look at the number of eosinophils in the blood cell. Neutrophils stain a pale purple, and they have a lobe nuclei. Let's take a look at the picture here briefly. And pale purple, and see how their nucleus is divided into lobes? That gives the neutrophils their other name, polymorphonuclear leukocytes, meaning a leukocyte with multiple nuclear lobes. Any question about that? Polymorphonuclear leukocytes is such a long name that people often abbreviate it PMNs. I'll just call it neutrophils for short. They're also a motile and phagocytic cell, and as you can see, they're the most common white blood cell. They make up about 60 to 70% of the white blood cells in a normal person's blood. And a normal person would be somebody who doesn't have an infection and they don't have a worm infection or allergic response. But normally a person has mostly neutrophils as the white blood cells in their blood. They're important because they, as well as the eosinophils and the monocytes, can squeeze through the capillaries and get into the tissue where they'll actively move around in the tissue and destroy microorganisms. And the neutrophils are usually the first white blood cell that'll come to a site where there is a bacterial infection. And so in a bacterial infection, you can do a sample, what do you call it, a blood count at the infection. And if the patient has neutrophils as the main white blood cell at the site of the infection, the clinician can know that the patient is early on or early in the stage of having a immune response to the infection. Any question about that? Okay, so early on in the site of infection, you mostly have neutrophils, and we'll talk about the next one. Late in an infection, the neutrophils get replaced. In late in infection, you'll mainly see macrophages at the site of an infection, assuming the infection is in the tissue. Any question about any of that? So basophils, neutrophils, and eosinophils are granulocytes, and neutrophils and eosinophils are motile phagocytic cells. Now the third motile phagocytic cell is the monocyte macrophage lineage, and that lineage is agranulocytes, meaning the white blood cells without visible cytoplasmic granules. Let's show that picture again. Scroll that, let's move that over to the side there. So lacking the cytoplasmic granules in the monocytes, and they're, generally speaking, the third most common white blood cell in the blood. Obviously these two aren't that different, but this one's generally larger than the eosinophils, the monocytes. And we'll talk about monocyte macrophage, I should say. We'll talk about the difference there, but we're going to talk about those as one lineage, and in a sense they are one lineage. Did I confuse anyone there? Because I haven't talked about what we're talking about. They're the last motile phagocytic cell, and if you see the monocyte macrophage in the blood, it's called a monocyte. Some of the monocytes leave circulation, meaning they leave the blood, and then wander around in the body tissues. They are called macrophages if you find them in the body tissues. And if they're wandering around in the tissues, meaning they don't stay in one location, they are called wandering macrophages. Okay? And what a macrophage is, they're a little bit bigger than a monocyte, and they're a more mature cell. And they're a little more phagocytic, too, than a monocyte. So simply moving a monocyte into the tissues changes it into a macrophage, and the cell matures a little bit. Like I said, it becomes a little bit bigger. It's not much bigger, but on average, it's a little bit bigger. And it's a little more phagocytic. Any questions about any of that? So those were the wandering macrophages, and those are most of what the monocytes become. There are also some macrophages that are called fixed macrophages, and that's because they don't wander around in the body and they don't wander from one tissue to another. Okay? And certain tissues or organ have the fixed macrophages, meaning when the monocyte goes into this tissue or organ, the macrophage stays in that tissue or organ, like the liver or the skin. And they have different names for the different tissues, and I'm not going to go into the different names. What's surprising is that some of them look different than a macrophage, like the glioblastoma, a macrophage in the brain. It looks like somewhat like a brain cell, meaning it took on the appearance of the cells around it, and it's still a macrophage, but it's in the brain, and it has a different appearance than a macrophage in the liver, even though that's a fixed macrophage, or the macrophage in the skin. Did I confuse anyone on any of that? We're not going to get into the names, but each of the different tissues has a different name for the fixed macrophages. The important thing is the monocytes and the macrophages, because we'll talk about them as one cell lineage, are important phagocytic defenders. And I did mention that when a tissue gets infected, first come the polymorphonuclear sites, which I said I was going to call neutrophils, and that's the number one white blood cell that happens initially in an infection, and then later on the macrophages start dominating in the infection. So if a clinician does like a biopsy and says, you mostly have macrophages in the site of infection, that means you're well along in the host's immune response to the infection. Any question about any of that? All right. So the next and the last white blood cell we need to talk about is the lymphocyte. And let me remind you, this is not a leukocyte. It's not a phagocytic white blood cell of the blood. It's a white blood cell of the blood, but it is a specific immune cell, and these are the lymphocytes. Oops, I'm pointing at the wrong picture here. Sorry about that. It's the second most common white blood cell in the blood, and it makes up about 20 to 25% of the white blood cells in the blood. When you're looking at the lymphocytes, you'll notice there's very little cytoplasm. It's mostly a nucleus. And so when you look at these under the microscope, depending on how the cell is oriented, you might only see the nucleus. Like if the slide is only showing you this portion or this portion of the cell, you're only going to see the nucleus. You have to see it at the right angle to see a little bit of the cytoplasm, but it's mostly nucleus. Any question about that? I forgot to mention that the monocyte macrophage lineage stains purple, and the lymphocytes stain a light or pale purple. The lymphocytes are the smallest white blood cell, and they're involved in specific immunity, the third line of defense, which we will talk about in our next lesson. Any question about the white blood cells? Okay? So let's talk about phagocytosis, which happens with the neutrophils, mainly the phagocytic cell, the monocyte and the macrophages, also a very phagocytic cell, and the eosinophils, which can be phagocytic, but remember they can play a role in an allergic reaction, and they can also play a role in defending the host from a helminth infection, but they can be phagocytic. Okay, so let's watch a little video on...
Here, under the microscope, you can see a white cell. The large, shapeless body in the middle, surrounded by red corpuscles. It's about to engulf that long, thin body, which is an invading bacterium. Watch. If there's a large-scale invasion, we may need antibiotics, but our white cells give us a great deal of protection.
Okay, any questions? That was a very good video of phagocytosis. There's the bacteria being engulfed, and right about there, it's not quite totally taken in a phagosome inside the white blood cell. Now, what do you notice which is unusual, close enough, about this video? This is the white blood cell, probably a neutrophil, and there's the bacterium. What's unusual about this video? Nobody wants to take a guess? What size is the most bacteria compared to like a red blood cell, which is pretty close to an average size of a human cell? The white blood cell is a little larger than the average human cell. What size is a bacterium? Come on, people. We've talked about this before. Nobody's gonna guess. So I'll try calling on people. Dana. Or Dania. What's unusual about that bacteria? No comment. Yolanda, what's unusual about that bacteria? Angeline, what's unusual about that bacteria?
I would just say it's very long and it's one single bacteria. I don't know.
Yeah, it's much longer than a bacterium because most bacteria are about 1/10 the size of a red blood cell. And that is actually much longer than a red blood cell. It's thinner than a red blood cell. There's only one bacterium I know which is this long. And my guess is whoever made this video specifically ordered this very big bacterium so that you could see phagocytosis much more readily when he videoed it and then put it on YouTube. Okay? Because that's much bigger than any bacterium you would ever see if you were to go into a lab. Or whenever you look at the pictures of the bacterium that I will show you. That's much bigger than any bacterium you'd ever see. Like I said, there's only about one bacterium that I know of that's that size. And that's the point I wanted to make. This is a very unusual bacterium because it's so large. But thank you, Angeline, for answering. Phagocytosis by neutrophils, eosinophils, and monocyte macrophages occur in the 5 phases we're going to go through here. First, there's chemotaxis. Second, there's adherence. So the phagocyte has to adhere to the invading microbe. Third, there's ingestion. Fourth, there's killing of the microbe. And then fifth, there's elimination. Any question about any of that? All right, so let's talk about each of them. Maybe I'll put this at the bottom. First, there's attraction, the phagocyte, and remember that can be 3 of the white blood cells, has to be attracted to the microbe and then move towards it. Most of the time, the phagocytic cell will be in the blood and the infective agent will be in the tissue, so the phagocyte has to leave the blood and be attracted to the invading microbe. And here we're seeing, let me get that out of the way, a white blood cell being attracted to a bacteria, and it's moving towards the bacterium. And what happens is this white blood cell put out a pseudopod here, and it'll pull up its membrane on this side and put more of the cytoplasm here, and so this will expand and grow, and the cell will eventually move from this position to that position. And that's how a white blood cell moves, by what do you call that? I just said it, putting out its cytoplasm here, pseudopods, yes, it puts out a pseudopod here. All right, any question about that? And it doesn't tell you how it's being attracted, but chemicals from this cell or a damaged normal human cell are released by this cell or by the normal damaged human cell, and those chemicals attract the phagocytic white blood cell to move this way. And what the white blood cell does, it just moves up the gradient for the concentration of the chemicals. So as you get closer and closer to this cell, you'll increase the concentration of the chemicals that the white blood cell is using to move to the to the invading microbe. Any question about any of that? Now, what was I going to say about that? Oh, yes. Besides chemicals being released by the invading microbe or a damaged human normal host cell, there can also be chemicals released by other white blood cells, especially the B cells, and also by complement proteins, and we'll talk about the complement proteins later in this lesson. They're a protein they move around in circulation, and they can attach to the invading microbe and then attract white blood cells to the region. And that chemicals, the complement proteins, and the antibodies released by a B cell, they can coat the invading microbe, and that coating is called the chemical coating, meaning what's doing the coating, is called an opsonin, and it attracts white blood cells to the region. And the coating process is called opsonization, which attracts white blood cells to the region. Did I confuse anyone there? I sort of stumbled a little bit on that. Anyways, that's the process of attraction, which the white blood cell is being attracted to the invading microbe. Okay? Any questions about any of that? So once the white blood cell is attracted to the invading microbe, and I forgot to mention that that attraction is called chemotaxis. The white blood cell is finding chemicals and then moving up the chemical gradient to be attracted to the invading microbe. Once the white blood cell arrives at this location, it then has to attach to the pathogen, and that is what we call the stage of adherence. The phagocyte has to adhere to the microbe. Now, phagocytes can adhere directly to the microbe unaided. Any question about that? But a white blood cell can be aided by an opsonin protein. So the opsonins sort of do two things, and that is they can attract the white blood cell to the region, and then they act as a bridge to adhere the white blood cell to the phagocyte. Okay? Any question about that? And let me show you how they can act as a bridge. I wonder why I didn't have a picture for that. Here we go. So this antibody is doing something different we're not going to talk about, but the antibody binds to the pathogen, and then that can cause a white blood cell, which that's actually not a white blood cell, but can cause a white blood cell to bind to the antibody, and then the antibody can act as a bridge, binding the white blood cell to the pathogen. And I think I know why I'm not talking about it now, and that is I'm going to talk about it in our next lesson. And that is how a white blood cell can adhere to a pathogen. Okay? Any question about that? All right, the next phase of phagocytosis is ingestion, and that starts by the white blood cell putting out pseudopods around the pathogen, and then these pseudopods just grow, and then they fuse together, bringing the pathogen inside the white blood cell, inside a phagosome. Any question about that? And that's called ingestion, where the pseudopods surround the pathogen and bring it into the white blood cell in a phagosome. And here we're looking at a phagocyte engulfing a cell that is almost as large as the phagosome, and it's putting out pseudopods and will slowly engulf that cell. Obviously, the smaller the cell, the more easily the phagocyte can phagocytize it. Any question about any of that? Next, what happens is the digestion or killing phase. What happens is the white blood cell brings a lysosome up to the phagosome, and then they fuse together. And then the digestive enzymes of the lysosome, which is called a phagolysosome when they fuse together, the digestive enzymes of the lysosome are released into the phagosome, and then they start digesting the pathogen. There's about 30 enzymes in a lysosome. We've only mentioned one, and it gives the lysosome its name, and that is lysozyme, an enzyme that digests peptidoglycan. But together, there's about 30 enzymes in the lysosome. And the lysosome is also fairly acidic, around pH 4 from lactic acid. And so the acid is not good for the human pathogen. And there's also a bunch of noxic substances in the lysosomes, like hydrogen peroxide, superoxide radicals, hydroxyl radicals. And the point of all these oxygen radicals is that if the lysozyme and the enzymes don't digest the pathogen, the oxygen radicals will bind to the pathogen and then kill it. Okay? Any question about any of that? Once the phagolysosome is digested, meaning the pathogens inside it are digested, it's no longer called the phagolysosome, it's called the residual body, and that's sort of a phagosome with digested parts of the pathogen. And then that phagosome, or residual body, moves up to the cell membrane and then fuses with the cell membrane, which then allows the contents of the residual body to be expelled from the cell. And that gives the fifth step of phagocytosis, elimination, meaning the white blood cell just dumps the residual body or the contents of the residual body out of the cell. Okay? Any question? Oh, and that happens by exocytosis. The residual body has a membrane, and that membrane just fuses to the cell membrane, and then this is open and the contents leave the cell. Now what happens if this is a gram-negative cell? What's going to be released right here? Let's say we have a precious brain cell right here, or a precious heart cell. What's going to be released right there if that's a gram-negative cell? Come on. What's in a gram-negative cell? Something noxious. You guys are really not talking today. Okay, Ian, what's in a gram-negative cell that's something noxious? That this cell right here, that's a precious brain cell or a precious heart cell, would not want to be exposed to? No answer. Layla, what's something noxious in a gram-negative cell? Okay, Angeline, what's something noxious in a gram-negative cell?
I have no idea. Killing microbes by enzymes and other chemicals. The chemotaxis is the only thing that I know that's just more of a vehicle or movement. But I don't know.
Okay, thank you for answering, but no, that's not right. How about an endotoxin in the gram-negative outer membrane, lipid A? When that's digested, that's going to be released in the process of elimination. And then this cell right here, it's going to have to deal with that. And so hopefully that's not a precious brain cell or a precious heart cell, because it's going to be exposed to that endotoxin. And it's just something you need to worry about in Phagocytosis. A gram-negative cell always has a problem, and the host is going to have to deal with it. Okay? Now the white blood cell is going to want to digest that gram-negative cell, because you don't want that growing and free to grow in the body. But even when you kill it, you're going to release the endotoxin, the lipid A, from the outer membrane of the gram-negative cell, and then that's going to be released by the white blood cell. And then the neighboring cells are just going to have to deal with it. Okay? All right, that's just the point I wanted to make. I mean, it could be that this cell produces other noxious substances. We haven't talked about other toxins, but there are other toxins besides endotoxin. And this cell here is going to have to deal with it. Of course, the other toxins could be released by this microbe anyways. So exotoxins tend to be released by the microbe. The endotoxin is the only one which is not released until the cell dies. And that's because the endotoxin is a part of the outer membrane of a gram-negative cell. So the cell does not usually want to release that endotoxin, because you'd be releasing the lipid A. All right, any questions about phagocytosis? There's a few things we need to talk about phagocytosis, and that is a few pathogens actually will live inside the cells of a phagocyte. For example, chlamydia and Mycobacterium tuberculosis and the malaria parasites, they can all live in a phagocyte. And what they do is they will actually reproduce in the phagocyte. What they do is they prevent the fusion of the phagosome and the lysosome. Okay? And in case the two do fuse together, the pathogen also keeps the pH of the phagolysosome high enough that the enzymes do not work, meaning the enzymes work at best around pH 4, and if the pH is off from that, like around pH 7 or higher, then the enzymes are not going to work, or at least not work well. And so they'll keep the pH too high for the digestive enzymes to work. Any question about that? Another thing is, I forgot to mention that the white blood cell will inactivate the oxygen radicals in the lysosome. So that is not released. The white blood cell will inactivate those. And one way of inactivating it, I'm not going to talk much about that because the white blood cell will inactivate each substance, but I'm only going to talk about how it inactivates hydrogen peroxide. Is it brings in the enzyme catalase into the residual body, and the catalase destroys the hydrogen peroxide. So then what is released will not be hydrogen peroxide. Okay? So what was in the lysosome will not be released to the neighboring cell. Only what came from the pathogen might be released if the white blood cell doesn't neutralize it. But it neutralizes all of the oxygen radicals. Chlamydia, Mycobacterium tuberculosis, and the malaria parasites, like I said, can live inside the white blood cells. And the main way they do that is they prevent the phagosome, the lysosome, from fusing, but they have backups too. They keep the pH so that the enzymes in the lysosome don't work. A few leukocytes can kill extracellularly, where the white blood cell, oops, lost that, what did I move? Doesn't phagocytize the pathogen. It'll come up to the pathogen and then release substances on the pathogen to kill the pathogen. And that's called extracellular killing, where the white blood cell binds to the pathogen and then releases substances extracellular to kill the pathogen. And that actually is the normal mode of action of eosinophils. They'll come up to a helminth, bind to the helminth, and then secrete chemicals extracellularly on the helminth, and then that will start killing the cells of the helminth. Okay? And that's the normal way that eosinophils kill, at least to kill a helminth, because the helminth is too big to be phagocytized. And then it's also the way killer lymphocytes, we'll talk about that in our next lesson, they will come up to a cancer cell or a virally infected cell, and they will release chemicals up on that cancer cell or the virally infected cell to kill the pathogen extracellularly. Any question about any of that? All right. I can't see anyone today, so hopefully you guys are doing well on understanding the lesson. All right, let's move on to inflammation. Inflammation is a localized, non-specific response from tissue damage, and the tissue could be damaged by a large number of substances such as heat, chemicals, mechanical damage, or microbial infection. When inflammation is used against a microbial infection, the process of inflammation is to confine the agent of injury, destroy the agent, trying to show you my hands, and then repair the damage. Okay? The symptoms of inflammation are the area will have redness, there will be heat and swelling, and then everyone's favorite symptom, pain. Okay? So those are the symptoms of inflammation. We're going to talk about inflammation happening from a puncture wound, but it can happen in any of these different responses by heat, chemicals, mechanical damage, or microbial infection. So I'm going to talk about it happening in a puncture wound, which will then get infected. Although the puncture itself will cause the inflammation because the tissue will be damaged by the puncture. When cells are damaged, they release inflammatory chemicals, and there's a number of them. Bradykinin starts the process, but histamine, kinins, prostaglandins, and leukotrienes are all chemicals that continue the inflammatory process, and they sort of continue the process that bradykinin started, and histamine is the main one we'll talk about. What it does, all these other chemicals increase or continue the process that bradykinin and histamine started. What histamine does when it's released, it increases the vasodilation and the permeability of blood vessels, and that increases the flow of blood to the region, and that increases the plasma fluids into the tissue, because the permeability of the capillaries in particular increase, and so more fluid can move into the tissue. That can bring chemicals as well as cells to help defend the area, and then it can bring in the chemicals needed to start building repair, meaning start repairing the area. Any questions about any of that? So both processes, vasodilation and permeability, can increase the flow of white blood cells to the area. They'll arrive at the site of injury and deliver antimicrobial chemicals, as well as phagocytize any infective pathogens. And that will, with the increase in the flow of blood to the region, will increase swelling in the area, as well as warm the area up, possibly making it red, and it can cause pain in the region. So let's look at vasodilation. This is a picture of the normal permeability of actual vanuoles, and then a little bit of capillaries right there. And then after histamine increases vasodilation, hopefully you can see that that's actually the vanuoles and the capillaries are much bigger. They're swollen to increase the flow of blood to this region. Any question about that? Now, what happens with a vanule and a capillary is that normally the cells of the wall of the vanule or capillary, they have gaps in it for fluid, the plasma, to leak out of the blood and then flow into the tissues. Well, after histamine increases the permeability of the vanules and the capillaries, so that there's much greater space in between the cells of the wall of the capillary or the vanule, and so much more flow of fluid moves into the tissue, and that will increase the pressure and the swelling in the tissue. Any question about any of that? Now, as a side, by increasing the size between the cells of the wall of the vanule or the capillary, that will also make it easier for a white blood cell in the blood to leave and enter the tissues. Okay? And so right here we're seeing a monocyte squeezing out of the blood cell or blood circulation and moving into the tissues. And once it moves into the tissue, it'll become a macrophage. All right, I've already stated that the kinins, the leukotrienes, and the prostaglandins, really what they do is intensify and continue the effect that histamine started. And that will attract white blood cells to the region where there's infection. Also, with the increase of the flow of blood to the region, we'll have more blood clotting proteins move to the wound, and so you can have a clot forming. I already talked about that. The purpose of the clot is to engulf the region where there's pathogens in the clot, and that can keep the pathogens walled up so they cannot spread and move to other areas of the body. Any question about that? Now, the bad thing about a clot is that if it gets into the blood, if it's in the tissues, it's fine. But if it's in the blood, the clot can break off and then circulate in the blood. And if a clot hits the lungs, it can cause a clot in the lungs. And you need a lot of them, but it can do damage to the lungs. And you can, if you have a whole bunch of clots hitting your lungs, you can impair the lung's ability to breathe. And you start doing damage to the lung. The lungs is actually a better place for the clot to hit because the lungs can take one or two clots. If a clot happens in the brain, it causes a stroke, and if the clot hits the heart, it can cause a heart attack. Any question about any of that? Okay. Pus may form in the wound area as the host cell dies and fluids from the cells are being released. And then the bacteria start dying and they'll release their fluids, and that can add to the pus. And then the phagocytes can come into the region and eat so many cells that the phagocytes die, and they will add to the pus. And you can have the walled os area, and that can form a cavity or an abscess. Inside an hour of the site of infection, you'll have neutrophils start moving into the region and start accumulating at the site. They actually are attracted to the damage as well as the invading microbes, and what they'll do is they'll move to the region, moving with the blood, and they will to the region where there's pathogens. They will stop moving in the blood, and they'll be attracted to the endothelial cell lining, and that's called immigration, where they'll stop and then start moving through the wall of the venule or the capillary, and then start moving into the tissue. And when they move into the tissue, that's called immigration, moving from the blood into the tissue. The process of margination and immigration just takes a couple of minutes for the white blood cells to move out of the blood and into the tissue where the invading microorganisms are. Monocytes will follow neutrophils. Once monocytes move into the tissue, remember we call them macrophages, and if they wander about in the tissues later, they'll be called a wandering macrophage. So neutrophils will be found at the site of inflammation initially, but later on macrophages will start dominating. Remember, macrophages are a bigger and more phagocytic cell, and besides phagocytizing the pathogen, the monocytes can also start phagocytizing the dead tissue and the dead cells of the host, meaning they're a little bigger and better at phagocytizing, and they'll start cleaning up the region where the infection is. The white blood cells, both the neutrophils and the macrophages, if they eat too many things, meaning they phagocytize too many things, they themselves will die. And if they die, they will add to the pus of the wound. And then that pus, when it starts accumulating, it'll either ooze out on the surface, like on the surface of the skin, or it will be slowly absorbed by the body, so that the pus will be eventually disappear from the the site of the infection. Okay? Over time, the damaged tissue will be repaired. And what's surprising is the repair process actually happens during inflammation while the body is still fighting off the infective agent. So repair begins once inflammation begins, and there will be repair proteins there to start the cells rebuilding, and the tissue will eventually repair itself, okay? And that repair process usually happens after the pathogen has been defeated by the host immune system. Okay, any questions about any of that? All right, I'm showing you a picture of the inflammation from a puncture wound, and during this puncture we're getting an infection of bacteria, the damaged cells as well as the invading bacteria release chemicals which attract white blood cells to the region, and the chemicals also start the inflammation process. We'll have increased permeability and vasodilation so that more fluids will be released in the area, and that can cause swelling, warmth, the heat of the area, and redness. Pain, if the swelling is too great, the swelling will cause the nerve to feel pain. The increase of the blood flow to the region will bring first neutrophils and then macrophages to the region, and they'll come in and start phagocytizing the invading microbes. And also in the area, the repair proteins and repair cells will come in, and they'll start repairing the damage from the puncture wound as well as the damage from the bacteria. And we will get accumulation of damaged tissue forming pus. We could get clotting in the region, which will wall off the area so the bacteria cannot get out of this region and invade the blood and then go elsewhere in the body. And eventually the bacteria will be fought off, and assuming the patient survives, and then the repair process will continue, and the puncture wound will be replaced, and the damage will be repaired. Any question about any of that? So during infections, during the course of an infection, the frequency and the number of white blood cells can change in the blood. Clinicians monitor both the total white blood cell count and the count of the various types of white blood cells, and that can be an indication of what step of inflammation and infection the patient is in. Like I said, early on in infection, neutrophils will dominate. Late in the stages of infection, the macrophages will dominate. Fever is another nonspecific host immune response to an infection. Fever may inhibit the growth of some microorganisms. That's why we have an elevated temperature. It makes the bacteria, the elevated temperature, grow at a suboptimal temperature, meaning each bacteria has an optimal temperature that will grow out, and a fever makes the temperature for the bacteria to grow suboptimal. Okay, so that's one of the things of the fever. The fever also intensifies some host defenses and speeds up the repair process. Simply getting a fever, let me blow that down so you can see the whole picture. Simply getting a fever will increase the flow of blood throughout the body, increasing the flow of white blood cells to the region where there's an infection, increasing the repair proteins and stuff like that. Any question about any of that? So that's how the fever intensifies some of the host defenses and speeds up the repair process. It's been shown and then clinically experimented also that if you let the fever run its course, the patient will be sick for less time than if you use fever-suppressing medication. Meaning if you take fever-suppressing drugs, you will be sick for a longer period of time than if you do not take the fever-suppressing drugs. For that reason, some clinicians refrain from using fever-reducing drugs unless the fever is too high or too long. Obviously, if the temperature is too high, you can get damage from the fever, so you don't want the fever to be too high. However, other clinicians say that the benefit of having a fever is too slight and the fever is too horrible to endure, and so these clinicians will always give a fever-suppressing drug. Okay? So it's obviously a difference of opinion as to whether you should give fever-suppressing drugs. If you have a child who has a fever, my advice to you is, as long as the fever is not too high or too long, do not give the child fever-suppressing drugs, because when they feel awful, they're less likely to get into trouble. Any questions about that? That was supposed to be a joke, by the way. All right, so we've talked about phagocytosis, inflammation, and fever as a secondary line of defense. There are two other secondary lines of defense, and those include interferon and complement, which alter the course of an infection. And I want to check my time here to make sure I'm not going to go over. All right, so actually we're going to be stopping here because it's close to 2:15, and I don't want to start the next process. And we still have another day. Actually, we've got 2 more days, don't we? No, tomorrow's Thursday, so we have 1 more day to talk about host defenses 1. So this is a good place to stop. So are there any questions? If there's no questions, I'll see you at 2:30, and I think I'll just be asking questions about the lab or other questions because I don't think you have a lab today. Okay? All right, so I'll see you later.